Howdy, everyone. Um, so we are done with talking about sort of general activities in cells, including how they use energy and how they change, how they modify different types of energy, transfer it from one form to another. Okay. Um, where we're moving next is to focus on inheritance and genetics and how DNA functions. Okay. Um, and so there are several different topics that all kind of fall into this under this category. As always, like to do things in a little bit different order than your book does. Um, where I like to start is I like to start with Mendelian genetics. Okay, so I like to start with genetics because for a variety of reasons. First of all, I think that by talking about this first, it helps you appreciate a little bit more about like why you should care about DNA, right? So it's like when we talk about, how you inherit all of these different traits that you have that make you unique, right? Um, that kind of helps you appreciate why we need to learn this other stuff about cell division and about DNA and how it works. Okay, so that's the way I like to do it. So we're actually going to be doing chapter 5 sort of backwards, right? So this week we're going to do the second part of chapter 5. Next week we're going to do the first part of chapter 5, right? And then we'll be moving on to DNA stuff after that. Okay. Um, so let's jump into this. So once again, I'm going to have videos that are kind of broken down by topic. So the first thing we're going to do is kind of talk about the history of genetics a little bit, and some of those sort of real basic stuff. Okay, some of this might be somewhat familiar, especially if you are younger and you have fairly recently you have memories of high school biology, right? You might remember some of this stuff. If you don't, that's absolutely fine. In fact, sometimes it's better because then I'm dealing with more of a clean slate, right? One of the things I want to warn you about. So we're going to start with like the basics, right? And then we're going to get into sort of more complicated topics within this area. Okay. One of the things I want you to know about genetics is that unlike a lot of other topics that we've covered in this class, so most of the time when we talk about stuff in this class, it's like, here's this concept. I need you to understand it. I need you to have examples. I need you to be able to explain examples, all that kind of stuff. Genetics is very much more, I mean, that's also true for genetics. But in addition to that, we're also going to be doing a lot of like problem solving, right? So there's a question, right, where it's like, mom has this, dad has this, what are the chances of baby having this, right? Questions like that. And so as a result of your need to be able to do genetics problems, you have a lot more homework on this topic than usual. And I expect that you will probably want to work on some of this a little bit. In Zoom meetings, right? Because some of this, it just requires practice, right? And I have a million, practice questions up my sleeve. So come talk to me, right? And we can work through this, okay? I'm just going to show you a few examples in the lecture today, mostly because I'm expecting that you're going to come to Zoom to get some help, and or you're going to be working through your homework and kind of figuring out how to do this stuff as you're doing your homework. Okay. All right, so let's talk about genetics. Let's talk about inheritance. So um it's so funny, people for a long time, people didn't know what the heck they were talking about. Right. And so one of the kind of early thoughts about how babies were made is that sperm contained like a tiny little human in them, and that's where babies came from. Okay. um I find this interesting because on the other hand, also it was very common for women to be blamed as the responsible party when a child they were carrying was female instead of male, right? So in most cultures, right, because patriarchy is a thing, you know, it, back in the bad old days, hopefully not now, okay, but back in the bad old days, it was often preferable to have sons instead of daughters, right? And so, yeah, women were sometimes, divorced, or in some really weird cases, especially with like royalty, murdered, right? Because they didn't produce an heir for the king that was male. Anyway, we'll talk about why that's a bunch of hooey at the end of this chapter. Okay. So people didn't know what the heck they were talking about for a really long time. Okay. The father of modern genetics is Gregor Mendel, and he was alive in the mid 1800s. I want to say he was born in like 1820 something, right? So, he was an adult doing research in the mid 1800s, and he was a monk, right? So just like Darwin, one of the interesting things about both of these gentlemen is that we've talked before about this sort of perception that a lot of people have that like science and religion are like enemies, right? But most biologists at that time, right, were religious folks, right? So the reason they were driven to understand nature was from the sort of motivation to explore God's creation, is how they thought about it, right? And so Mendel was a monk, right? And he spent his days tending a garden with pea plants, and he did a ton of research on inheritance of all kinds of different characteristics in pea plants. So there's a couple of images of my homie Greg. Yeah, and there's a picture of a pea plant. Some of you, if you've taken biology before that you recall, a lot of the examples in intro bio classes talk about what Mendel studied, which are the pea plants, right? But now all the gen bio textbooks are like, nobody cares about peas. So they're switching all their examples to other stuff. So most of the examples in your book now are about dogs, which is more fun. Okay. So anyway, but we are going to talk about the pea plants a little bit. So the reason that Mendel studied pea plants is they're easy to grow. They're common. They had traits that were very clear and delineated. And it was really easy to like control reproduction in pea plants. Okay. So in general, let's go back to this picture here for anatomy of a pea flower. All right. So we haven't talked much about plant anatomy at this point, and we're not going to talk too much more about this other than this. But essentially, for most flowers, the way that they reproduce is most flowers have both male and female reproductive structures. Not all, but most of them. Sorry. Ugh, almost sneezed, but then I didn't. They have male and female parts. Okay. So the female part, typically called the carpel, has eggs, contains eggs, and the male parts, called stamens, contain sperm, right? They have pollen, which essentially pollen is like a little package that contains sperm. All right. And so, many plants, not all, but many plants are pollinated by a pollinator like bees or butterflies or birds that carry pollen from the male structures on one flower to the female structures on another flower. Okay. The cool thing about pea plants, and one of the reasons why they were so useful for Mendel, is that it's really easy to control where the pollen goes if you want to, right? So he could easily take pollen from one flower and dust it onto the female reproductive structure on another flower, right? He could cut off the stamens from one flower so that it wouldn't. That interruption necessitated a long enough gap that I have no idea what I was talking about. Based on the image, I'm going to guess that I was talking about pea flowers and how you can move pollen from one plant to a specific plant, right? And right, so if you're interested in, say, I want to know what kind of offspring would this plant and this plant produce, right? You can, using a little paintbrush, just pick up some of the pollen and put it on the specific plant, excuse me, that you want to mate it with, okay? The other thing you can see in this picture is you can see them cutting the stamens, that's the male parts, off of this plant so that it doesn't accidentally self-fertilize, okay? So, yeah, so that's how he actually, you know, that's the dirty work, right, of actually doing these studies. Now, one of the benefits of peas, aside from it, they're easy to grow, they grow fast, they're, you know, easy to keep in captivity and all of that. They have a lot of characteristics that are very like straightforward, either this or this, right? So when you're trying to study these different traits in peas, you can study their color, the flower color, the position of the flower, the color of the seeds, the shape of the seeds, the pod shape or color, the stem length, right? All different kinds of things. And so that's what Mendel did, right? And Mendel came up with a lot of the terminology that we use today. Now, one of the things that I find really interesting about Mendel is that he was doing all of this research around the same time that Charles Darwin was as well, right? They were definitely contemporaries. And the reason I find that interesting is that Darwin knew, like, he was like, I know that evolution has something to do with things that are inherited, right? But he didn't really understand the mechanics of how inheritance worked. Mendel did, right? And so I often wonder in my, you know, in my nerd imagination, I often wonder, would we be sort of farther along in our understanding of all of this stuff if Mendel and Darwin had crossed paths, right? If Darwin had read some of Mendel's work, would he have been like, oh, that's what, right? And then he could have, you know, called up. No, he couldn't have, because they didn't have phones. He could have written a letter and, you know, had it carried. Dude, take a month to get there. To Mendel, being like, yo, your inheritance stuff, that's cool. Check this thing out that I'm working on, right? And they could have, like, you know, collab. I don't know. I'm just saying, would have been interesting. Okay, so when we talk about Mendel, the father of genetics, just like Charles Darwin is the father of evolution. When we talk about Mendel, we talk about two of his laws, okay? And so we're going to kind of illustrate those as we go through and talk about this stuff, okay? So here's the first one, all right? So what we're looking at here is we are looking at basically a progression of inheritance in flower color. Okay. So when we talk about genetics, and we don't do this a lot in this class, but just so you're aware of the terminology in case you run into it. Sometimes we talk about there being different generations. So we talk about the P generation, the F1, and the F2. Okay. So P stands for parental. Okay. F1 is first filial. So basically what that means is the offspring of the parents. Okay. So in this particular example, if Mendel started with a plant that had purple flowers and a plant that had white flowers, and both of them were true breeding, meaning that if they self-fertilized, so if the white flowered plant self-fertilized, because remember, they have both reproductive structures on their flowers, right? So if they were allowed to self-fertilize, okay, the white flowered plant only made more white flowered plants. The purple flowered plant only made more purple flowered plants, okay? So if our P generation was a true breeding purple and a true breeding white, what he found was 100% of the time, if he carefully bred those two plants together, that purple and that white, all of their offspring, 100% of them would have purple flowers, period. Okay? All the time. The F2 generation basically is if we take that F1 generation, so we take the offspring and then we cross them with each other, right? Then you end up with the next generation of plants. So try not to think about this too much because it gets sort of icky. Because it's like, if these are the parents up here and then these are the, you know, brothers and sisters down here, and then they have babies together and that makes the grandchildren, ew, they're plants, y'all, it's okay. Right? So F2 is like the second, you know, generation of offspring, right? So it's the grandbabies, okay? So the F2 generation, if we crossed the F1 generation, right? So we crossed F1 with other F1s. What you ended up with is what Mendel ended up with is a very reliable ratio where 3/4 of the offspring would have purple flowers and 1/4 would have white flowers. Isn't that interesting? What's the deal with that? Right. And so eventually he saw these patterns enough that he developed some symbols and some terminology to kind of track all of this. Okay. So this is that same exact story, but now we're adding symbols to it. Okay. All right. So Mendel's first law, by the way, is called the law of segregation. Okay. And basically what Mendel was able to figure out is he was able to figure out that all of these different traits that he was examining are inherited by what we now call genes. Okay. We've talked about this a little bit before when we were talking about evolution. So, you know, you've heard that term before. So a gene, remember, is basically a sequence of, oh, oops. Like I do that every time. Okay. So a gene is a sequence of DNA that codes for something. Okay. And what Mendel was able to figure out is that all of these plants, all of these individual plants actually had 2 genes. So they didn't just have one, they had 2. And those genes could be identical to one another, or they could be different. This is where the term allele comes in. So what an allele is an allele, and by the way, I hate editing this section of the transcript because when I say allele, the auto transcript thing, thinks I'm saying illegal. Makes me mad. Anyway, so an allele is basically different versions or different types of a gene. That's not very legible, sorry. Okay. All right. So in the previous example, right? So we have our purple flowered plant and we have our white flowered plant. Okay. So the gene that we're talking about here is flower color. Okay. So there is a gene that is responsible for flower color. So gene for flower color. Right? Well, that gene has 2 different versions or 2 different alleles. Okay. And so in this particular example, right, those 2 versions of that gene are either purple or white. Okay. So gene is like all the different possibilities for a specific trait. Okay. And then alleles are the different possibilities, the different versions of that gene. Okay. So, and remembering that Mendel discovered that, you know, each individual has 2 genes, which may or may not be identical. Okay. So the purple flowered plant, we're symbolizing here with 2 capital P's. The white flowered plant, we're symbolizing here with 2 lowercase p's. Why? Okay. So it is conventional, meaning this is what most people do, that some of these traits, some of these alleles are dominant over other alleles. Okay, let's unpack that for a second. Okay, so if we're looking at our P generation, right, our parents, we have a purple flower plant, we have a white flowered plant, but all of their babies, all of them are purple, right? What that means, the terminology that we use to describe that phenomenon is we say that the purple flowered allele is dominant to the white flowered allele. Okay, so the white flowered allele we say is recessive. All right, so purple is dominant. And if you need reminders of this stuff, we'll define those terms in one sec. Okay, right here on this little table, they're listed, right? So purple is dominant to white, which is recessive. Okay, having yellow seeds is dominant to having green seeds. Okay, all right, now let's talk about those words dominant and recessive. Dominant does not mean better. It does not necessarily mean more common. Literally all that it means when you have a dominant allele and you have a recessive allele is that if both of the alleles, both of the versions of the gene are present, then the dominant one is what you're going to see. The recessive one, it covers up the recessive one. Okay, so we call that dominant. All right. Okay. What else are we looking at here? All right, so the thing that Mendel was able to figure out, the reason this is called the law of segregation, okay, what he discovered is that in these adult plants, right, that had either big P, we use uppercase generally for dominant, that's conventional, and then we use lowercase, but the same letter for the recessive, okay? So we're using P to stand for purple, right? And so big P is purple, little p is white. Okay. Why do we use little p? Why not use W? I'm pausing so you can think about it for a minute. Well, we want to remember that these are not completely separate genes, right? These are the same gene. They're genes for flower color. There are just different alleles of that gene. So we want to use the same letter. Yeah. And so we go with the letter that, you know, is for the word that is the dominant allele. Okay. So that's why we use P. All right. That's conventional. There's exceptions. Whatever. Okay. All right. So the adult plants have 2 copies of their alleles, okay, of their genes. The thing that Mendel was able to figure out, and he didn't know why, because he didn't learn anything about cell division. That's where we're going next. After we talk about genetics, we're going to go to cell division. Okay. He didn't know anything about that. But what he did know is he was able to figure out, well, somehow those 2 things separate from each other, right? So those 2 genes, right, those 2 alleles for the flower color separate. And so the gametes, gamete, remember, is a generic term for eggs and sperm. Contain only 1 gene, not 2. Okay, so if you have 2 big P's, if those are your 2 genes, those are your 2 alleles, right? Then your gametes, your eggs or sperm, will only have big P's in them because that's all you have, right? But each one only has 1 big P. Okay? Because of meiosis. We're not going to talk about that yet. We're going to talk about that later. Okay? The mechanism of how it happens later. Okay? For our white flowered plant, right? They have 2 little p's, 2 of the recessive alleles. Okay? When those separate in gamete formation, right? Each gamete then has a little p because that's all they have. Okay. Now, in reproduction, what happens? You got an egg, you got a sperm. they come together. We call that fertilization, right? So one gamete comes from this parent, the other gamete comes from that parent, and there's our F1 generation. Now, we know that from this parent, right, they got a big P. And from this parent, they got a little p. So we know that these plants have both a big P and a little p. And they appear purple because purple is dominant over white. God, I got interrupted again. Now I don't remember where I was. Okay, so the purple in the F1 generation, they all appear purple because the purple allele is dominant to the white allele.
It feels like an absolute brick.
Awesome. Thanks, honey. Perfect. Sorry. This is what happens when I try to do online teaching. It's a white hot mess. White hot mess. Okay, so the entire F1 generation, all of those plants have one big P and one little p. Okay, so back to this whole law of segregation, right? Remember that when our F1 generation, when they produce their gametes, right? Their gametes are going to have only one. But in this case, because they have both a big P and a little p, half of their gametes are going to have a big P and the other half of their gametes are going to have a little p. Sweet. Okay. All right. Now we're going to breed the F1s together, right, to come up with our F2 generation. Okay. And so this is where we run into something called a Punnett square. Okay. So a Punnett square is a tool that is used to predict the ratio of what kinds of offspring you can produce. Okay. Now, this is not like a checklist. Okay. So this is not saying that literally if you have this F1 generation and you make exactly 4 babies, 3 of them are going to be purple and 1 of them is going to be white. It doesn't work like that. This is like a ratio. This is like a percentage. This is probability. This is not a promise. Okay. So it's not like a checklist. It's a probability. Okay. So how do we set up a Punnett square? How do we deal with a Punnett square? Okay. This is setting up a Punnett square is the key to being able to do genetics problems. Key. Okay. It's not challenging if you just remember what the purpose of the Punnett square is. Okay. And like how you're supposed to set it up. Okay. Because if you set it up wrong, then your answer is going to be wrong. Okay. So in this particular example, they're showing you sperm on sort of the top, and they're showing you eggs on the side. It doesn't matter to me if you swap those. So I don't care. You don't need to have the male gametes on the top and the female gametes on the side. I could not care less. Either way works just fine for me. I don't care about that. Okay. All right. So what do you write on the sides of the square? Well, essentially what you're writing is you're writing, well, what could those gametes contain, right? So if you've got a male flower in this F1 generation, remember that half of his sperm, on average, are going to carry a big P, and half of his sperm are going to carry a little p. So we put a big P and a little p. Okay. All right. Now, if we're looking at a female of this generation, right? Half of her eggs are going to have a big P. Half of her eggs are going to have a little p. So we put a big P and a little p. Now, once again, it's conventional to list the dominant ones first. That's usually how it looks. Does it have to be? No, it still will tell you the same thing if you do it differently. So if you put your little p first and then your big P after, it would still ultimately tell you the same thing. It's just that's not usually how people do it. They usually do the dominant first. Okay. And then all you do is you just fill down and across. Okay. So I take my big P sperm and I go big P, big P. I take my little p from the sperm and I go little p, little p. I take my big P from the egg and I go big P, big P. And then I take my little p from the egg and I go little p, little p. Okay. All right. Let's real quick. Let's do a white screen for a sec. Okay. So let's say that we were crossing. I'm going to do the exact same thing that we were just looking at. We're just going to, okay. We're just going to do it without the picture. Okay. So if we are doing a cross where we're crossing one individual that has a big P and a little p and another individual that has a big P and a little p. By the way, the name for this, what we call this, is we say that these individuals are heterozygous. So heterozygous means, okay, so hetero means different, right? So heterozygous means you have two different alleles. Okay, so let's say that, your instructions are you have two individuals that are heterozygous for, this trait. Okay, and you want to know what are all the possible outcomes that you could see in their offspring. All right, well, this individual, what letters could they have in their gametes? Remember, the gametes separate. Right? So there was two alleles, but we're separating them because that's what happens in gametes. Okay. So they could have either big P or little p, right? How about this individual? What could they contribute? Same. Big P or little p. All right, that's okay. That's cool. And then the world's ugliest square. And then, like I said, we just fill down and we fill across. Okay. So those are the big P's that I'm dragging down. These are the little P's that I'm dragging down. And then across, big P, little p, little p. Okay, cool. All right, now remember that P, right? So an important thing about genetics is showing your work, right? Now, on the test, how much you're going to be able to show your work? You're not really, okay? But you do have a scratch pad that you can use in Canvas, okay? So you can show your work while you're doing it, right? I just can't see it, okay? So big P, remember, was purple, right? And purple was it dominant or recessive? Dominant, okay? And then white was recessive, so it's a little p. Okay, so now what I want to know is, now that we've done this, right? What I want to know is what kinds of offspring can they have, right? So first of all, what did these two parents look like? What did these guys look like? What color flowers did they have? Well, because they're heterozygous, right? Meaning they have one dominant allele and one recessive allele, they express the dominant trait. So they both look purple. Okay. All right. Okay. So what would this individual look like? Two big P's. Right. Well, let's introduce some terminology. So homozygous means, if heterozygous means two different alleles, what does homozygous mean? Two of the same allele. So typically when we say homozygous, we then describe whether or not they're homozygous dominant, meaning that they have two of the dominant allele, or homozygous recessive, meaning they have two of the recessive allele. Okay, so that individual, that possible combination that they can produce, homozygous dominant, what would they look like? What would their appearance be? Well, they have two big P's, so they'd be purple. Okay, now this square and this square, both of them are, let's use our terminology, heterozygous. So what does that mean, heterozygous? Right? They have two different alleles. They have both the dominant and the recessive. What would they look like? They have both purple and white alleles. They have both. But what would they look like? That's right. They would look purple because purple is dominant over white. Okay, so they're heterozygous. Now, what about this? That's two little p's. So first of all, is it homozygous or heterozygous? Homozygous, because it's two of the same. And we would probably say homozygous recessive, because it's two of the recessive alleles. Yeah? Okay. Now, what would the appearance be? What would that plant, what would their flowers look like? Would they be purple or would they be white? They'd be white because they have two recessive alleles. Yeah? So remember, dominant doesn't mean better or worse. Dominant doesn't mean more common or less common necessarily. All dominant means is that it covers up the other allele, right? So the purple covers up the white when you have a heterozygous, okay? So in heterozygotes, I'm zyg because I ran out of room. Okay, the dominant allele covers up the recessive allele. That's what it means to be dominant over recessive. Okay. All right. So remember what I said a second ago. This is not like a situation where it's like, this is a promise. So if we get exactly 4 offspring, one of them is going to be homozygous dominant, 2 of them are going to be heterozygous, and 1 of them is going to be homozygous recessive. This is not a promise. This just tells you probability, right? So there's a 25% chance or a 1/4 chance that any offspring that they are producing would be homozygous dominant. There is a 1/4 chance, right, 25% chance that their offspring will be homozygous recessive. And there is 2/4 or 1/2, right, 50% chance that they'll be heterozygous, which means they will appear purple. Okay, another set of terminology. Okay, so we got dominant and recessive down. We got that, I think. We got heterozygous and homozygous. Sweet. We know the difference between a gene and an allele. God, there's so much vocabulary. Okay. The last thing we need to talk about are the terms genotype and phenotype. Okay. So genotype means what alleles they possess. Okay. So which alleles? Okay. So one way of describing the genotype is using these terms, right? So if I wanted to describe the genotype of this individual, right, I would say its genotype is that it is homozygous dominant. Or I could say its genotype is 2 big P's. Okay, so genotype is all about what alleles do they have. Okay, phenotype is all about what trait is expressed. Which trait is expressed. Now, the way that people often, including myself, talk about this is we say, what do you see? Well, just keep in mind that not all characteristics are visual, right? So in this particular example, it is visual, right? This plant either has, I can see it either has a purple flower or has a white flower, so it's visual, right? But some characteristics you can't tell just by looking at an individual. Okay, so we talk about the phenotype is what you see or what it looks like, but in reality, what we really mean is which trait is expressed, right? And so in this particular case, right, what trait is expressed for this guy? Purple. How about these? What trait is expressed for those two? Also purple. Okay, so if we are to talk about something called a genotypic ratio or a phenotypic ratio. Now we're looking at probability of each of these different combinations. Okay, so rather than write this, I'm going to go back to this because it's written out. Okay, so here's our Punnett square. We filled it. We did exactly the same thing. I just wanted to go through it together slowly. Okay. All right. So phenotypic ratio and the genotypic ratio. I wrote them on opposite sides. Sorry. Right. So our phenotypic ratio. Remember, phenotype is what's expressed or what you see with the air quotes. Look at my air quotes. Okay. The phenotypic ratio of a breeding of the F1 generation, right? So the phenotypic ratio in the F2 generation is a 3-to-1 phenotypic ratio, three purple to white, 1 white. Okay. The genotypic ratio, now remember genotype is sort of more specific, right? Because it's what actual alleles do they have, not just what they look like. Okay, so the genotypic ratio is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Okay, so that's the genotypic ratio. Okay, so Mendel was able to figure all of this out just by doing crosses with pea plants. Okay, and he did enough of them that he could figure out, first of all, that all individuals for all of these different genes that he looked at, all of these different traits that he looked at, in these examples anyway, he discovered that there's 2 different kinds of alleles that are found in every individual. When that individual makes their gametes, their eggs or their sperm, it's only one. So they split in half, right? Half of them end up in, some of the sperm, half of them end up in the other sperm. Half of them end up in one egg, half, right? And then when fertilization happens, we're recombining. So 2 are coming back together. Yeah. So he was able to figure, that's essentially what the law of segregation is, right? Okay. Right. Now, in your book, they're talking about different traits, and they spend a lot of time. I can't remember exactly what there's some stuff in the first section that I'm like, why are you spending all your time talking about this? I don't care about this. Some traits are really, some characteristics are really straightforward like this, right? Where it's like either purple or white, real straightforward. Only one gene is responsible. Other things are much more complicated, right? So the character of eye color in humans is actually more complicated than this, right? It's not like you either have this or this, right? There's a range of eye colors, right? Some people have super light blue eyes like this person, right? Some people have real dark blue eyes. Some people have kind of blue-green eyes or blue-gray, right? There's all different shades of brown from very dark to very light. And then there's hazel, which is like brownish, goldish, greenish. And then there's green eyes. Once again, there's a range in colors of green, right? There's people who have eyes that are almost gray. I mean, right? There's all this variation. Eye color is not inherited super simply like flower color in pea plants is. Okay. So that's just important thing to keep in mind. Okay. All right. Here's a little graphic that talks about what an allele is. I think we got this. I think we're good, right? But look at it. Okay. Here's a little graphic that talks about the terms heterozygous and homozygous. Remember those? Okay. So we're good. So one thing we haven't talked about yet is we haven't talked about something called a homologous pair of chromosomes. Okay. So homologous chromosomes. Pair that contain the same genes. It'd be good if I could spell genes. Let's try that again. Okay, so in this particular example, and we haven't talked about the structure of DNA yet much. Okay, so just work with me for now. Okay, essentially we got 2 chromosomes here. So chromosome is basically a strand of DNA. Okay, this is a homologous pair of chromosomes, meaning that this blue one and this red one, they're colored that way just so that you can differentiate them. Okay. They carry the same genes. Whether or not the alleles are the same is a different issue. Okay. So in this made up example, this homologous pair of chromosomes has two different genes. It has the B gene and it has the D gene. Usually there's a ton of genes on every pair of chromosomes, but like thousands. Okay. But in this one, it's just two. It's B and D. Now, they're not necessarily identical. They don't necessarily have the same alleles, but they have the same genes. Okay. So for the B gene, it turns out in this example that they happen to be heterozygous, right? So the chromosome that came from dad is dominant. The one that came from mom is recessive. Okay, whatever. So the individual whose chromosomes these are is heterozygous, right? For this particular gene, right? They inherited a big B from one parent, a little b from the other parent. Sweet, they're heterozygous. They happen to be homozygous for the D gene. They got dominant from both parents. Okay? All right, so that's homologous chromosomes. Okay. We already talked about genotype and phenotype. Awesome. Look at that puppy. Ugh, they use labs in instead of pea plants because nobody cares about pea plants. They use puppies instead. Look at that. That little puppy is like looking into my soul and being like, I'm the cutest thing you've ever seen. Okay. Anyway. Okay. So yeah, so they're just showing you relationship between genotype and phenotype, right? For a few different characteristics. So it just so happens that having freckles, right? And I don't mean like the random age spot here and there. I mean like proper freckles. Like she has proper freckles all over the place. Okay. Having proper freckles. Freckles are a dominant trait. That doesn't necessarily mean common, remember? All it means is that when a trait is dominant, when an allele is dominant, it just means that it covers up the recessive, right? So I think I'm trying to think of all of your profile pictures. Right? I don't think there's any of you guys that have freckles, or a very small proportion of us in this class have freckles. Okay. So just because it's dominant doesn't mean that it's more common. Okay. All right. So because she has freckles, she's homozygous dominant, right? Because she's got two big F's. Okay. There's our sweet little Labrador puppy, right? Well, it turns out that in Labrador retrievers, their coat color, it's actually way more complicated than this, but for your book's purposes, right? Black is the dominant allele, and that chocolate brown color is a recessive allele. Okay. So if you have a puppy that has a chocolate brown coat, that is a recessive trait. So that tells us, because it is a recessive trait, just by looking at this puppy, I know that this puppy is homozygous recessive because I can see that he's brown. Okay. And that's a recessive trait. So that means he has to have two of them, because if he had the dominant allele, it would cover up the recessive allele, right? More about this. Well, if you're like, what? Don't worry, there'll be more of this. Okay. And then here's a purple pea plant. Okay. So just by looking at that purple pea plant, purple flowered pea, I can't tell just by looking at it if it's heterozygous for the flower color allele gene, for the flower color gene, or if it's homozygous dominant. I can't tell just by looking at it because it's purple. Right? So it could be homozygous dominant. In this case, in this example, they're showing you it's heterozygous. Okay, so this is just kind of helping you understand genotype, phenotype. What does it mean to be homozygous dominant, homozygous recessive, heterozygous, blah, 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 blah. Okay, so it's kind of reviewing the stuff that we've been talking about. Okay, here's a graphic from another book that kind of walks you through the law of segregation. The idea is that alleles separate, right? So each individual has two alleles. They separate when you make your gametes. And then when gametes are fertilized, when gametes come together, we're back to having a pair. That's all it means. He didn't even understand like the mechanism of this. He didn't know anything about meiosis. Meiosis is the type of cell division that produces gametes. You don't know about it either. And part of the reason why I like to actually talk about genetics before we talk about meiosis is because Mendel didn't know about meiosis. They didn't even have like good microscopes then. They didn't know what DNA was. They didn't know any of this stuff, right? So when he was talking about traits, he's like, well, there's something that's inherited, but he didn't know what it was or what it looked like or how they separated or any of that, right? He saw a pattern and he was like, well, I know this pattern is happening, right? But he didn't know like visually, like he didn't know structurally what was actually happening. So I kind of figure, hey, why not talk about genetics when we're talking about Mendel with the same body of knowledge that he had, which is we don't know anything about DNA yet, and that's okay. Okay, so that's why we do the second half of chapter 5 first. And then we go back and do the rest of chapter 5, we'll be like, oh, that's why Mendel got this, that. Oh, that's the explanation of why. Yeah, so we're doing the what first, and then we're going to do the why. Okay, so that was his first law. Oh God, this is turning into a very long video. Okay, I'm going to try to keep it shorter. Okay, so here is a super adorable example of another Punnett square. But now instead of looking at boring old pea plants, now we're looking at beautiful doggies. Okay. I find it irritating that the two parents are just the same picture that's flip-flopped. Okay. We're just going to have to pretend that this is daddy and this is mommy. Even though if you look carefully, the picture is the same picture. It's just flip-flopped. You think that they could spend, $5 more to get a picture of another dog. I'm just saying. Anyway. Okay, so here they're setting up the Punnett square, right? So we want to know if we have this black dog, dad up here, mom down here, right? If we've got a papa lab that is black, but he's heterozygous. He's got a big B and a little b. And we've got a mama who's also a black dog, but she's also heterozygous, right? Okay. We want to know what kind of puppies can we get out of this, right? So here's our little key. And by the way, this is something that I, whenever I'm working through a genetics problem, I always write all these little factoids down, all these little tidbits, right? So why did we pick B for this allele, right? If we're talking about the colors black and chocolate, why did we pick B? Well, because we always choose for our symbol, right? We always choose the dominant allele. And so big B represents the black allele, and then little b represents the chocolate allele. I think I didn't finish my thought earlier because I got interrupted. Maybe I did. I don't know. I might be repeating myself. Sorry. Why do we use little b instead of C for chocolate? I did tell you this, right? We want to remember that, in fact, these are not separate genes. These are all coat color genes. They just are the two different alleles, the two different versions of that gene. Okay. So remember, the way that we set up our Punnett square is always the same. We put, what are the combinations of the gametes, right? So from dad, he can produce sperm that have either the big B or the little b because he has one of each. From mama, she can have eggs that have either a big B or a little b because she's got both, one of each. Okay. And then we just fill down and fill across like we did before, right? So wait a minute. This is a mistake. I love it when I find errors. Okay, look at that. That's not right. We got a big B here and a big B there. So that puppy is homozygous dominant. Okay, I wonder if the mistake is in your book or just in the PowerPoint. I'm gonna actually look because I'm curious now. Let's see. Is the mistake in your book or did they actually yeah, see, they actually proofread the book, but apparently they don't proofread the PowerPoints. Whoever made the PowerPoint screwed that up. Anyway. Okay. Well, I made this is anyway, I take the pictures from the publisher, right? I don't make these things myself. Okay. Right. So this guy, look at that face. Don't you just want to kiss it? Well, anyway, super cute puppy. Look at that fat little puppy belly. Okay, sorry. right? So this guy has a big B and a big B. So how would we describe him? What would we say his genotype is? Well, we could say it's big B, or we could say it is homozygous dominant. Okay, now this box gets a little b from dad and a big B from mom. So the genotype is heterozygous. This one also is heterozygous. Big B, little b from mom, big B from dad, also heterozygous. And then this little sad little face, little b from dad, little b from mom, two little b's, must have the recessive phenotype. So homozygous recessive. So his coat color is chocolate instead of black. Okay, so what is our genotypic ratio? Well, there's three different genotypes, right? This is one genotype. This is another genotype, and that's the third genotype. So I don't like where they call this the results. Let's call this the genotypic ratio. Genotypic ratio is a 1 to 2 to 1. Okay, now I've said this before, but I'm going to say it again. This is not like a checklist or a promise. So it's not like, okay, these two dogs mate and they have exactly 4 puppies, and you are going to end up with one that is homozygous, 2 that are heterozygous, and 1 that is homozygous recessive. No, that's not how it works. Each puppy has a different probability, right? Each one has a probability of ending up with this genotype or this genotype or this genotype, okay? So each puppy is an independent event. So rather than like thinking of it like a checklist, think of it as like there's a 25 for any puppy that is produced, there's a 25% chance that it's going to be homozygous dominant. There's a 25% chance that it's going to be homozygous recessive. And there's a 50% chance that it's going to be heterozygous. Okay. That's the way I want you to think about it. Okay. And then our phenotypic ratio down here, 3 black to 1 chocolate. Okay. So if you're a breeder and you're in the market of trying to breed chocolate labs because there is a bias against black dogs, it's a real thing, people. It is a real thing. Black dogs are less likely to get adopted in shelters than dogs of other colors. It's true. Okay. All right. So let's say that you are a Labrador Retriever breeder, but you want chocolate labs. You don't want too many black labs because those are harder to get adopted, right? To sell. Okay. So would you want to, if you were trying to get as many chocolate labs as possible out of this, would you want to breed 2 black labs together? Well, probably not. There's a possibility if they were both heterozygous, there's a possibility that you could get a chocolate lab out of the mix, right? But 75% of the time, you're going to get a black lab with this cross. Only 25% of the time are you going to get a chocolate lab with this particular pairing. Okay, does that make sense? I hope so. All right. Law of segregation. There's the 2 chromosomes to separate, whatever. Okay. Oh, that's ugly. Okay. What do I want to do? Do I want to keep going? Yeah, okay. One more. Mendel's second law. Okay. So Mendel's first law was the law of segregation, right? Each individual has 2 copies of any given gene because you have a homologous pair. You have 2 homologous chromosomes that have those genes. Those chromosomes separate, so the genes separate, right? And the gametes end up with one or the other. Okay? Okay. Mendel's second law. It's called the law of independent assortment. You'll see that name in just a second. Okay. This type of cross that we've been looking at so far, all of these examples, right? All of this are examples of something called a monohybrid cross. Mono means one. So monohybrid means one gene. So we're only looking at one gene. In this case, we're looking at the gene for flower color. Now there's multiple alleles for that gene, but there's only one gene. Okay, sorry, another interruption. Monohybrid cross means that you're looking at one gene. There are two different alleles for this gene, but we're looking at only flower color. Okay. A dihybrid cross means now we're doing a cross where we're looking at two different genes. Okay. So in this particular case, okay, this is not in your book, by the way. Do I want to look at this? Oh God, there's more interruptions. Okay, so in this particular example for independent assortment, they're looking at two things. They're looking at seed color and seed shape. Okay, so for shape, sweetheart, I'm busy. I already told you I was busy.
Mommy?
Yes, Ellie.
I can turn it down to 0. I can turn it down to 0.
Okay.
You want to talk?
Okay.
By the way,
So
you are doing it.
they're looking at two different genes that are completely unrelated to each other. Okay. So they're looking at seed shape and they're looking at seed color. Well, if you go back to the chart at the beginning of the PowerPoint, we might as well. Let's do it. Whoa, whoa, whoa. Right? Yellow coloration is dominant to green. Round for seed shape is dominant to wrinkled. Okay. Let's go back to where we were. Whoa, whoa. God, I keep going out of control. Okay. So we choose R for shape. Why do we pick R? Well, our two alleles are round or wrinkled. Round is the dominant one, so we use big R for round, little r for wrinkled. Right? For color, yellow is dominant over green, so we use big Y for yellow, little y for green. Okay? I'm not going to explain the rest of this. I'm going to go with the example in your book. But what I want you to do with this is sit and think about it for a second. What's the deal that's happening here, right? Why do we get this? Okay. This is something that did not happen, which is why there's the big thing through it. Okay. Okay. This is what we're looking at. All right. So we're going to do the dog example that's in your book, right? But I want you to, take a minute to figure out why this happened. Okay. Why does your Punnett square look like that? Okay. So, all right. Now we are looking at, in our doggies, we're looking at two different genes. So we're looking at their coat color, so black or chocolate. And we're also looking at a particular type of hearing loss that's genetic. Okay. So we already know they use big B for black, little b for chocolate. It irritates me that they're using D for hearing, big D for hearing, and little d for deaf. Shouldn't they have used H? Because remember, normally what we do is we use the capital letter for the dominant allele. In this case, hearing is dominant. Deafness is recessive. So it's going against convention. Whatever. That irritates me. I'll survive. Okay. So here we have two homologous pairs of chromosomes, right? Here's the one that has coat color. Here's the one that has the deafness versus hearing gene. Okay. What Mendel's second law, the law of independent assortment, states is that these two genes are not inherited together, right? So they can be inherited in any combination. Yeah. So if this is our daddy here, and daddy is heterozygous for both, so he's got big B, little b, big D, little d. right? Sorry if you're dyslexic. This is going to be real hard with the B's and the D's. Okay. If he's heterozygous for both, what that means is that his sperm can end up with a bunch of different combinations. Okay. Because now we're talking about 2 genes instead of 1. You're going to have a B and a D in each sperm, one of each, right? In the dog himself, we have 2 B's and 2 D's, right? But in his sperm, he's going to have 1 B and 1 D. Okay, so what are the possible combos we can get from that? Well, you could have a big B and a big D. You could have a big B and a little d. You could have a little b and a big D, or you could have little for both B and D. Those are the 4 combos you can end up with, right? 4 combos. Okay.
Can you tell me if this is a side or a corner of?
Okay, so we figured out what would go on this for Dad, right? Oh, look, they've now managed to come up with a different picture of a dog. Okay, so we know, because he is heterozygous for both, we know that he has 4 different combinations of sperm. We're assuming that she is also heterozygous and she has 4 different combinations of eggs. Ellie, I'm ignoring you because I'm working.
I don't know what side is it.
Find your corners first. She's doing a puzzle. Anyway, okay. So she also has 4 different combinations. When you have a Punnett square that has more than 1 gene, like in this dihybrid cross, right? 2 genes. What you see, you end up with a 16 square Punnett square because you have more things to drop down and bring across, but it fundamentally works the same way. So we bring down our big B and our big D, and then let's keep the B's together and the D's together because it's easier to read, right? And then we have a big B and a big B, big D, little d. I'm checking it real quick to make sure they didn't make any mistakes in this. Ellie, stop whining. I'm going to kick you out if you keep whining because I need to work. I'm checking. You should check too. Okay, are there any mistakes? I didn't find any. Okay. But do one of these yourself, right? Practice filling it in and filling it across. Okay. So here they're saying results, but what are these results of? Is this genotype or phenotype that we're looking at? So it's a 9 to 3 to 3 to 1. 9 to 3 to 3 to 1 ratio. Is this a genotypic ratio or a phenotypic ratio? All right, well, there are 9 dogs that have a black coat and they can hear you when you yell at them. 1, 2, 3, 4, 5, 6, 7, 8, 9. And you're going to want to go through each one and check. Yes, he's black because he's got a big B. Hearing because he's got a big D. There's 9 of those. Okay, then there's 3 dogs that have a black coat, but they're deaf. Black and deaf. 3 that are chocolate, but they hear. Right? And 1 that is recessive for both. So the 9 to 3 to 3 to 1 is the genotypic ratio or phenotypic ratio? It's the phenotypic ratio. What happens when you record videos the day after Thanksgiving, the day after Halloween? See, I've already moved on. There's a lot of sugar in this house, which means there's some rage. There's some emotions. There's some highs and lows. Okay. So it's a phenotypic ratio, right? There's 9 dogs that have the phenotype of having a black coat and they can hear. There's 3 dogs that have the phenotype of having a black coat but being deaf. There's 3 dogs that have the phenotype of having a chocolate coat, but they can hear. And then there's 1 Now remember, once again, I need to remind you again, these are probabilities. This is not like a checklist. So it's not like, okay, if they have 8 puppies in one litter, it's going to be these. And then if they have 8 puppies in the next litter, it's going to be these. That's not how it works, right? It's a probability. So there's 1/16 of a chance that they will end up with a chocolate puppy that is deaf between when mating these two parents. Okay, that's what that's about. All right, that's independent assortment. That's Mendel's other law. Aren't they cute? Oh, I can't even handle it. Right? They're there. Okay. This is showing how independent assortment happens. Okay. We're going to talk about this more. For now, it's fine with me if you just appreciate the fact that we're dealing with 2 different genes and those are independent from one another. That's the like degree to which I want you to understand it at this point. Okay. When we talk about cell division though next week, then you need to kind of understand this. So we'll come back to this image most likely. Okay. All right. I'm just going to stop there because this is long. Okay. So I'm going to stop there and then this is where we'll pick up for the next stuff. Okay. All right. Stay tuned. Come back for the next video.